name is Eric Flemings, and this is Maluski & Company. Maluski & Company was founded in 2015 in Vallejo, California. Our company is named after one of our key mentors who was instrumental in establishing the vision and foundation of our corporation. He is a representation for the hard work, dedication, and commitment that each of our team members brings on a day-to-day basis. Here at Maluski & Company, we are very detailed in our approach to the domestic and global market. Part of that detail involves having a specific company vision, mission, and values. Our mission as an organization is to practice corporate responsibility by providing health and economic solutions to our customers, while also providing value to our key stakeholders. Our company values integrity, passion, innovation, and commitment to our customers, and we will not rest until we lead the way in cancer therapy and eradicate the spread of breast cancer globally. Breast cancer is considered triple negative when it is insensitive to estrogen, progesterone, and HER2. Because triple negative tumors do not contain receptors for estrogen, progesterone, and HER2, they are unresponsive to therapies that target those receptors. Because of this, treatment options are often limited, creating a need for new therapeutic solutions. Triple negative breast cancers tend to be more aggressive and have a higher mortality rate within the first 5 years compared to other forms of breast cancer. Rates of triple negative breast cancer have been shown to be highest in African American women. Currently, the treatment regimen for triple negative breast cancer involves surgery, radiation therapy, and/or chemotherapy. In addition to these therapeutic options, PARP inhibitors have been demonstrated to have efficacy against triple negative breast cancer. If we look at this timeline, we can see that PARP inhibitors initially came onto the market in 2014 and have been approved for the treatment of ovarian cancer and more recently, breast cancer. PARP stands for poly-ADP ribose polymerase, and it is a protein that is naturally produced in human cells. This protein helps to repair cellular DNA when it is damaged. In a healthy cell, this is a wonderful function. However, in a cancerous cell, this is a molecular target that can allow us to eliminate a growing tumor. Therefore, inhibiting PARP stops the tumor cell from being able to repair its DNA, leading to cell death. This brings us to Maluski & Company's latest product, liraparib, known as Torenza. The slogan for Torenza is, "A better life, a better journey," because as we will demonstrate in our clinical trials, our product is safer and more efficacious than any other PARP inhibitor on the market. Like all novel PARP1 inhibitors, it was designed using a thioimidazole scaffold. In our laboratories, we conducted a series of chemical reactions to arrive at our unique molecular entity, which through clinical trials has demonstrated the necessary properties warranting placement of our drug on all therapeutic guidelines for the treatment of triple negative breast cancer. Here is a brief overview of liraparib. As stated prior, our drug is a PARP inhibitor that is indicated for triple negative breast cancer. It comes as an oral pill formulation and belongs to the N-acylpiperazine class. The journey for this latest PARP inhibitor began in 2015 when we initiated and completed our preclinical trials, allowing us to continue on with our human trials. With that said, I will now introduce you to our clinical development leadership.
Our objective as the clinical team was to develop and perform a clinical development plan that determined the pharmacokinetic and pharmacodynamic profile of liraparib, and also to establish that it is a safe and efficacious alternative to the current standard of therapy.
Clinical development has been charted on this timeline. The preclinical studies were started in 2015, followed by clinical studies in 2016. Phase 1 and 2 clinical trials were conducted over the course of 1 year each, and Phase 2 trial took 2 years to complete.
The preclinical phase included in vitro and animal studies where we determined lethal toxicity of liraparib and demonstrated that it is sufficiently safe for testing in humans. Investigational new drug application was submitted and approved for clinical trials.
Phase 1 trial was an open-label, non-randomized dose escalation study.
The primary endpoint of this trial was to evaluate toxicity, pharmacokinetics, and determine the side effect profile of loraprib. The patient population for the NUEBRA trial was 23 women with triple-negative breast cancer with BRCA1 and 2 mutations refractory to standard therapy, which is conventional chemotherapy, with adequate organ function and performance status. The dose escalation design of the trial followed a modified Fibonacci sequence. We started with low dose equal to 1/10 of lethal dose of 2,000 milligrams, as determined in preclinical trials, and increased the dose gradually thereafter. Loraprib was given once daily as tablet formulation. The level 1 starting dose was 200 milligrams, level 2 dose was 400 milligrams, level 3 dose was 650 milligrams, level 4 dose was 950 milligrams, level 5 dose was 1,300 milligrams, and level 6 dose was 1,700 milligrams. The trial was started with a standard 3+3 design. A dose was given and 3 outcomes were possible. 1. If 0 toxic responses, the dose was escalated to next dose. 2. If 1 toxic response, it was continued with the same dose. And 3. If 2 or 3 toxic responses, the dosing was stopped since it reached maximum tolerated dose and went back to the previous dose. As the trial progressed, the dose escalation method was switched to time to event continual reassessment method to account for late onset toxicity. Toxicity was assessed using the common terminology criteria for adverse events version 4.03 at baseline, weekly during treatment, 5 times during the acute toxicity phase, and at all follow-up visits during the late toxicity phase. Follow-up visits in the late toxicity phase were done every 3 months until 2 years after the end of treatment. Patients were followed until the last planned study follow-up visit or until disease progression, whichever occurred first. Treatment response was assessed using a prone breast MRI 3 months after treatment. Toxicity in the NUEBRA trial was assessed in the following way. Dose-limiting toxicity was defined as nausea and vomiting grade 3 or greater in the presence of maximal support treatment, esophagitis grade 3 or greater, treatment discontinuation due to toxicity, dermatitis or rash grade 3 or greater, neutropenia, thrombocytopenia, and anemia. Maximum tolerated dose was defined as the highest dose level at which not more than 15% of patients experienced dose-limiting toxicity, or the highest reached dose level in the absence of dose-limiting toxicity is defined as the maximum tolerated dose. It was identified to be 1,300 milligrams. The pharmacokinetics profile of loraprib was identified as follows. The absorption is rapid, insignificantly altered by food intake. The distribution profile was the following. The volume of distribution was 130 to 135 liters and protein binding was 75%. Metabolism is primarily hepatic via CYP2D6. The majority of metabolism is through oxidation with some metabolites undergoing subsequent glucuronide or sulfate conjugation. Half-life was identified to be 8 to 13 hours and time to peak 1 to 3 hours. Elimination is through urine 35% and feces 52%. Among the adverse effects, the most common were fatigue, 35% of patients, nausea, 31%, vomiting, 23%, and headache, 22%. Rash and changes in mood were observed in 15% of patients. Hematological side effects such as anemia, decreased white blood cell count, and platelet count were seen in 12% of patients.
In the Phase 2 ENDURE trial, a prospective, randomized, open-label, multi-center study was conducted in which the current standard of care, chemotherapy, was compared with chemotherapy in addition to loraprib. The primary goals and endpoints of the ENDURE trial was a measurement of overall response rate to loraprib as well as progression-free survival. The secondary endpoint included patient-reported quality of life. A total of 123 triple-negative breast cancer patients with BRCA1/2 mutations were enrolled in this trial. Inclusion criteria for this trial included patients with triple-negative breast cancer with BRCA1 and 2 mutations, age 18 years or greater, histologically confirmed breast cancer, refractory to standard therapy, which is conventional chemotherapy, with adequate organ function and performance status. Exclusion criteria for this trial included those with patients with symptomatic or uncontrolled brain metastasis, uncontrolled infection, immunocompromised patients, patients currently having seizures or other severe illness, those who require treatment with inhibitors or inducers of CYP2D6, those who have received any investigational drug within 28 days of study entry, those with existing GI disorders that could interfere with absorption of the study drug, and pregnant patients. Patients were divided into 2 treatment arms: the loraparib plus chemotherapy combination treatment arm and chemotherapy only arm. Patients in the treatment arm received 800 milligrams of loraparib once daily on a continuous basis for 4 weeks until disease progression or other discontinuation criteria were met. Patients were seen at the beginning of each 4-week cycle for hematological and chemistry assessment, analysis of tumor markers, and physical examination and history. Imaging was done every 2 cycles, 8 weeks. Tumor biopsies were repeated at 8 weeks and at progression. For patients with toxic effects of grade 3 or higher related to loraparib, treatment was interrupted until resolution of the toxic effects to grade 1 or less. A maximum of 2 dose reductions were allowed, after which, if toxic effects persisted, the patient was withdrawn. Among the key findings of the ENDURE trial were overall response rate, progression-free survival, and patient-reported quality of life. The overall response rate in the chemotherapy and loraparib group was 83%, compared to 37% in the control chemotherapy group. Progression-free survival was recorded as 11.2 months in the chemotherapy plus loraparib group versus 3.3 months in the control group. For patient-reported quality of life, the FACT-B scale was used. FACT-B stands for Functional Assessment of Cancer Therapy for Breast Cancer. This is a questionnaire that assesses patients' physical, social, emotional, and functional well-being based on 37 questions. Nearly all items are rated on a 5-point scale, ranging from 0, not at all, to 4, very much. A higher score corresponds to decreased quality of life, and a lower score responds to an increased quality of life. In the ENDURE trial, the chemotherapy plus loraparib group had a score of 44.4 on the FACT-B scale, and the control group had a score of 111. These findings suggest that addition of loraparib to chemotherapy resulted in improved overall response rate by 46%, increased progression-free survival by 7.9 months, and increased quality of life by more than 2-fold.
Phase 3 trial was a prospective randomized double-blind multi-center clinical trial comparing the efficacy and safety of loraparib with a single-agent chemotherapy. The primary endpoints were overall response rate and progression-free survival. Our secondary endpoint was patient-reported quality of life. 1,323 patients with triple-negative breast cancer with BRCA 1 and 2 mutations were enrolled in the study. The inclusion criteria were patients with triple-negative breast cancer with BRCA 1 and or 2 mutations, age 18 years or greater, with histologically confirmed breast cancer, refractory to standard therapy, with adequate organ function and performance status. The exclusion criteria were patients with symptomatic or uncontrolled brain metastasis, uncontrolled infection, immunocompromised, currently having seizures or other severe illnesses, required treatment with inhibitors or inducers of CYP2D6, received any investigational drug within 28 days of study entry, existing GI disorders that could interfere with absorption of the study drug, and pregnancy. Patients were divided into 2 treatment arms, the loraparib treatment arm and chemotherapy only arm. Patients in the treatment arm received 800 milligrams of loraparib once daily on a continuous basis. Treatment cycle was 3 weeks until disease progression or other discontinuation criteria were met. Patients in the control arm received protocol-specified single-agent chemotherapy of the physician's choice. capecitabine, eribulin, gemcitabine, or vinorelbine, in continuous 3-week cycles. Hematological and chemistry assessment, analysis of tumor markers, physical examination, and history were monitored every 3 weeks. Imaging was done every 2 cycles, 6 weeks. Tumor biopsies were repeated at 6 weeks and at progression. For patients with toxic effects of grade 3 or higher related to loraparib, treatment was interrupted until resolution of the toxic effects to grade 1 or less. A maximum of 2 dose reductions were allowed, after which, if toxic effects persisted, the patient was withdrawn. Among the key findings of the lead trial were overall response rate, progression-free survival, and patient-reported quality of life. The overall response rate in the loraparib group was 86.9% compared to 44.5% in the control group. Progression-free survival was 22.7 months in the loraparib group versus 13 months in the control group. For patient-reported quality of life, FACT-B scale was used once again. In the lead trial, the loraparib group had a score of 24.9 on the FACT-B scale, and the control group had a score of 124. In conclusion, loraparib resulted in a significantly longer progression-free survival and in a nearly double the overall response rate than standard of care chemotherapy. In addition, loraparib has shown to significantly improve patients' quality of life as compared to chemotherapy.
Industry pharmacists are valuable members of the clinical development team. These pharmacists have basic foundational knowledge of molecular structures and their resulting structure affinity relationships. They understand the importance of establishing pharmacokinetic and pharmacodynamic profiles of drug therapies. They understand safety and efficacy profiles and the use of evidence-based medicine to prove the value of drug therapies. They also have patient interaction skills. On a clinical team, pharmacists can demonstrate their value by overseeing the clinical program while ensuring that patients are receiving the best care that our clinical team can offer throughout the clinical trials. Next, let's talk to the regulatory affairs team.
Our job as the regulatory affairs team is to get loraparib to the market. We orchestrate the strategy for the preparation, review, and submission of the appropriate documents to the FDA and other regulatory entities.
Regulatory affairs actions have been charted on this timeline. FDA interactions occurred prior to preparing the investigational new drug application, after phase 2 clinical trials, and prior to preparing the new drug application. We pursued a fast-track designation for loraparib, so that communication with the FDA would be more frequent, expediting its approval for the drug market.
The goal of the pre-IND meeting is to receive feedback and support from the FDA on the IND filing strategy, including the discussion for fast-track designation. Loraparib qualified for fast-track designation based on its ability to treat a serious condition, and it also addresses the current unmet medical needs for the treatment of triple-negative breast cancer.
With recommendations from the FDA, Regulatory Affairs prepared an investigational new drug application, which included Forms 1571 with a request for fast-track designation, 1572, and 3674. Key messages of the application supported loraparib's qualification for the fast-track designation as a drug that can address the current unmet medical need for efficacious treatment of triple-negative breast cancer, with its greater efficacy and lower incidence of hematologic side effects compared to existing therapies on the market.
Coinciding with the conclusion of the Phase 2 clinical trial, we requested an end-of-Phase 2 meeting with the FDA to evaluate the current body of clinical trial data and seek guidance on how to proceed with the safety and efficacy trials in Phase 3 to ensure meaningful outcomes. We then met with the FDA once more for a pre-NDA meeting in preparation for filing a new drug application for loraparib to discuss clinical trial endpoints, manufacturing, indications, labeling, and a timeline for submission.
The finalized NDA package consisted of the forms listed here, as well as all studies, data, and analysis from animal and human studies that support loraprib's superior safety and efficacy to existing triple-negative breast cancer therapies. In addition, FDA reviewers were provided with adequate information to assess labeling, appropriate manufacturing, proposed indications, sale, and distribution of our drug. Once submitted, an NDA application can take 6 to 10 months to review, and reviewers may request additional information or changes. At all steps of this process, our regulatory affairs team did our utmost to comply with FDA requests to ensure that the NDA application was accurate and complete.
Industry pharmacists are invaluable in the realm of regulatory affairs, with their intimate understanding of drug therapy, ability to analyze and interpret clinical data, and perspectives on real-life clinical application. Pharmacists can powerfully articulate what a new drug can contribute to the existing market.
Their drug-focused expertise can inform strategies for developing clinical trials and collecting the evidence needed to make a strong case for approving new therapies that can make a difference in the field of healthcare and the lives of patients. Next, let's talk to the marketing team.
Our objective as the marketing team was to develop a commercial strategy to differentiate loraprib from other PARP inhibitors on the market and to highlight its stance as the safest and most efficacious alternative to the current standard of therapy.
Now that we have completed our clinical trials, we are in phase 4 of our marketing timeline, where we will join the medical affairs team in showcasing our product.
Our positioning statement for loraprib is as follows. Loraprib, the most convenient and efficacious PARP inhibitor on the market, promises significantly lower adverse effects than the next leading treatment and can be trusted to responsibly provide the safest solution for patients fighting triple-negative breast cancer. We surveyed patients, and across the board, we found that the two most desired drug characteristics for PARP inhibitors were minimal or no adverse side effects and optimal efficacy. Providers also shared similar desired drug characteristics, but also wanted more indications and less drug-drug interactions. Additionally, we surveyed payers about their desired drug characteristics, and we found their most desired drug characteristics was decreased cancer recurrence.
Current FDA-approved alternatives on the market include the following agents, per the 2020 National Comprehensive Cancer Network Guidelines. Following the initial launch of loraprib, we don't anticipate tremendous or exponential growth, but within 5 years, we expect that our medication will share the market with the current leading PARP inhibitor. Projections show that within the next 15 years, loraprib will dominate the market based on our reputation as the safest, most efficacious, and least expensive PARP inhibitor. We look forward to exploring partnerships with local nonprofits, such as the Breast Cancer Prevention Partners, to provide genetic screening for patients who may be a carrier of BRCA1 and 2. With partnerships such as these, we intend to impact the local community by screening patients and possibly even diagnosing their cancer early before it's had the chance to progress. During these screening events, we will collect data and share relevant information about the value of our product. We are excited by the prospect of partnering with the National Football League every October as they demonstrate their support for breast cancer patients. Malusky and Company will join their efforts while emphasizing the value our drug can bring to patients suffering from BRCA1 and 2 breast cancer. Pharmacists' plethora of drug information expertise would contribute to accurate, educational, and effective marketing materials. Pharmacists have clinical experience and knowledge that would allow them to interpret and evaluate data for marketing purposes. Pharmacists are also knowledgeable in how to match drug therapies with individual patient need and would be influential in strengthening and conveying that point in our marketing materials. Pharmacists are trained to communicate to and with various audiences, a valuable skill when determining how best to share our company message. Now, let's see what Medical Affairs has been up to.
The role of the Medical Affairs Department is to provide scientific and clinical support for the new medication, loraprib. Some of our key activities are managing relationships, working with the data, and publishing reports from clinical trials, while presenting educational information and providing product safety information for healthcare professionals and patients. Medical affairs often plays a role after a phase 3 clinical trials that have yielded encouraging results. Once we knew that our drug was likely to come to market, we began strategizing the many ways that we can bring awareness to loraprib's new arrival onto the scene. Here at the Malusky & Company, we take an integrated team approach. The Medical Science Liaison team collaborates closely with the Medical Information team and Medical Communication and Publication team. The unifying factor is the data we have generated as a company. Medical Science Liaisons are one of our disease and drug experts. It is their responsibility to network in the field and provide the latest information about the value loraprib brings to triple-negative breast cancer treatment, as well as collect information about possible competitors that may be coming through the pipeline. Medical communication and publications are responsible for publishing our data and creating materials that can be handed out to the public. They also play a key role in helping to train our sales team. Our medical information team is responsible for relaying any relevant drug information to internal and external stakeholders. Anytime somebody has a question, they will be connected to a medical information specialist who can provide them with the answers. Part of our medical affairs strategy is identifying key opinion leaders. At the moment, some of the key opinion leaders in the field of breast cancer include Dr. Jason Rozick, who is an assistant professor in the Department of Melanoma Medical Oncology at the University of Texas Anderson Cancer Center in Houston, Texas. Another potential leader in the field is Lorenzo Haluzzi, who currently is an assistant professor of cell biology in the Department of Radiation Oncology at the Weill Cornell Medical College. Elizabeth Middendorf is another thought leader who is currently a co-director of breast cancer clinic research at the Dana-Farber Cancer Institute. Another aspect of our company is to ensure that we are present at all conferences related to breast cancer therapy. Some of these conferences include Breast Cancer 2020, located in Berlin, Germany, the San Antonio Breast Cancer Symposium, and the Lynn Sage Cancer Research Foundation. By networking at these conferences, we will be able to identify more key opinion leaders, as well as give information about loraprib and why it should be first-line therapy for the treatment of triple-negative breast cancer. Here at the Malusky & Company, we understand the power of digital revolution, and we aim to remain ahead of the technological curve. One of the partnerships we wish to explore is the Grail Incorporated. We believe that we can propose a breast cancer initiative partnership with this company in order to utilize their genetic data collection in order to spread awareness about triple-negative breast cancer and identify patients with BRCA1 and 2 mutations early before the onset of breast cancer. Another avenue we can explore is the creation of an innovative website that can streamline information to key internal and external stakeholders. We want to provide physicians with the most concise evidence-based medicine that they need in order to make decisions regarding their patients or their formularies. The value that the pharmacist brings in the medical affairs department is to provide scientific and clinical support for the new medication using those key information in order to differentiate our product from what is currently on the market. Pharmacists have the communication skill, which allows them to work well in a team environment. And using that, a pharmacist can engage in a doctoral level conversation and build relations in order to present educational information and safety information for healthcare professionals and patients alike.
On behalf of Malusky & Company, I want to thank you for your time and attention, and we are looking forward to having your support. Thank you.